Hello, class. Continuing on, let's go into the higher mental functions. All right, so we have cognition and language. Cognition, collective term for the diverse group of tasks performed by these association areas, remember, in the cerebral cortex. Right, so that in the cerebral cortex, the top layer of the cerebrum, gray matter, nuclei, cell bodies, dendrites, unmyelinated axons. Right, that's cognition. Cognitive functions, processing, responding to complex external stimuli, recognizing related stimuli, processing internal stimuli. We have cognitive processes responsible for social and moral behavior, intelligence, thoughts, problem-solving skills. We've discussed these different association areas of the cerebral cortex. Localization of cognitive function areas involved in cognition, parietal lobe, parietal association cortex, spatial awareness, focus on distinct aspects of specific object, temporal association cortex, that's near your temporal lobe, recognizing stimuli, especially such as faces. Prefrontal cortex, right? Frontal lobe, largest and most complex association cortices, make up a person's character, personality, gathers information from other association cortices, integrates information to give awareness of self, allows planning and execution of behaviors, right? You probably have heard of dementia, Alzheimer's disease. Patients with dementia have loss of memory, degenerative cognitive functions, changes in their personality, right? This is a brain disorder. We don't know how to prevent it. We don't know how to cure it. Some of the things that we can do are try to slow it, but we can't reverse it once it's started. Alzheimer's disease is just a common form of dementia. You get what we call these senile plaques, extracellular deposits of specific protein around neurons, degenerates the cortical neurons and synaptic connections. So the neurons don't connect as quickly, don't connect as well with each other. And you remember, the brain wants to communicate with other parts of the brain. It's all neurons. It's all nervous tissues, primarily neurons sending billions of signals to each other. And if they get degraded, you have loss of memory, loss of function, things like that. Right? That's what dementia is. Especially when it happens in cortical association areas and the hippocampus. Language. Right? Language, one of the most important functions of the brain. Able to comprehend, understand, what's being said to you. Like right now, you're listening to me. Also being able to speak, right? That's very important. Multiple brain areas work on this, as we've mentioned earlier. Broca's area in the frontal lobe helps produce language, allows you to be able to speak, planning and ordering words, putting them together. Wernicke's area in the temporal lobe helps you to understand language, linking words, understand what they mean. This aphasia is a language deficit, occurs when either of these two critical areas is damaged. Right, so that's an illness if those areas are damaged. Remember those, Wernicke's and Broca's area. You'll probably see those on an exam somewhere. All right, so the cerebral cortex, you see it here. Level of activity of neurons during speech. You see a lot of you see this Wernicke's and Broca's areas are working highly when you're speaking. Learning and memory, also very important. And this is where things can get a little difficult to understand. We have declarative fact memory, remembering that, anatomy is the form of the body, physiology is the function of the body. Those are facts. These are things readily available to consciousness, could in principle be expressed aloud. Then you also have non-declarative, which are procedural or skills, skills and associations that are largely unconscious. Like, for instance, you walk up to a door, you know to turn the doorknob. That's kind of an unconscious, non-declarative type of memory. Both can be classified by the length of time stored, right? So immediate memory, stored only for a few seconds. Short-term memory, stored for several minutes. Long-term memory, stored for up to a lifetime. The process of converting immediate or working memory into long-term memory involves a process called consolidation. You form these formation of storage and declarative memory appears to require the hippocampus, component of the limbic system, so different parts of the brain communicating with each other. Immediate and short-term memories are likely stored in this region. Your long-term potentiation, mechanism by which hippocampal neurons encode long-term declarative memories, seems to involve, and this is one of those areas where we don't know all the details, increase in synaptic activity between associated neurons. Although the hippocampus is required to form declarative memory, long-term memories are not stored in this region. It's stored in the cerebral cortex that correlates with their functions. Right? We change the brain that's how we get a long-term memory. Retrieval of these memories seems to be mediated by pathways involving the hippocampus and prefrontal cortex, part of the limbic system. Right? All right, so this is just some things about memory. Emotion, also interesting. Complex combination of three separate phenomena: visceral motor responses, which can be mediated by the hypothalamus. Your skin turns red, your heart starts beating. Somatic motor responses, smiling, laughing, frowning, things sort of on the surface. Feelings, highly subjective. All right, these are, we don't know exactly all the aspects of feelings. We know we have feelings. Obviously, everybody knows they have feelings, but we don't know exactly all aspects of feelings. Very complex, integrated with sensory and cognitive stimuli. You feel sad when you lose somebody, when you're watching a movie. What we seem to know is that the amygdala receives input from the brainstem, the thalamus, the cerebral cortex, the basal nuclei, analyzes these emotional significance of the stimuli and creates these associations and projects them to the prefrontal cortex. We don't exactly know how this all happens. That's what gives us feelings, which can change on a whim. Why do some people watch something and feel sad, and somebody else can watch it and not feel anything? It's amazing. The mind is very different in that category. All right, so how do we protect the brain? Let's go into that. Let's go into this protection of the brain. There's three features within the skull that protect the brain. Remember this. Remember, we have the skull. Very important. The skull is your skin. You have skin. If you have hair, that's your first line, and your skin, integumentary system. Then the skull is going to play a huge role in protecting your brain. But then after that, we have more protection: the cranial meninges, cerebrospinal fluid, and the blood-brain barrier. I always have trouble saying that for some reason. Right, so the cranial meninges, 3 layers of membrane surround the brain. 3 layers of membranes which surround the brain. You'll see these when you do dissections. You also have cerebrospinal fluid bathes the brain, fills the cavities. Remember the different cavities of the brain. And blood-brain barrier prevents many substances from entering brain and its cells from the blood. All right, so cranial meninges. 3 protective membrane layers, dense, irregular, collagenous tissue. Right, dense, irregular, collagenous tissue. Sounds like connective tissue. This is why the brain is an organ, has multiple tissue types. Structural arrangement from superficial to deep. We have epidural space, the gap, dura mater, subdural space, arachnoid matter, subarachnoid space, pia mater. Right, so you have gaps in between. There's dura mater, arachnoid matter, and pia mater. Right. Epidural space between the inner surface of cranial bones and outer surface of dura mater. Space is normally tightly bound to bone, only allows passage of some blood vessels. Dura mater, outermost meninges, meninx, you'll see the term used, thickest and most durable of the 3 layers. Double layered membrane, mostly collagen fibers, few elastic fibers. Periosteal outer layer attached to inner surface of bones of cranial cavity. So it goes right up to the skull. Functions as periosteum, extensive blood supply to epidural space. Also meningeal inner layer, which is avascular, superficial to arachnoid matter. You also see these dural sinuses, venous channels, cerebrospinal fluid, and deoxygenated blood from the brain's extensive network of veins, also where meningeal dural folds over itself. Courses between structures and brains. You have the subdural space, serous fluid space deep to the dura mater, below the dura mater. Houses veins that drain blood from the brain. You have the arachnoid matter next. That's the next structure. Second meningeal layer. Thin web-like membrane, dense irregular collagenous tissue. Remember, connective tissue acts as protector. Arachnoid trabeculae, collagen fiber bundles and fibroblasts, anchor arachnoid to deep pia mater, which is deep below the arachnoid matter. Arachnoid granulations, small bundles of arachnoid project superficially through meningeal dura into dural sinuses. Allow for that cerebrospinal fluid, return of cerebrospinal fluid to the bloodstream. We have another space below that, subarachnoid space. Major blood vessels of brain, also filled with cerebrospinal fluid. Then we have the pia mater, the last structure. So it's dura, arachnoid, pia, 3 of the streets. Those are the 3 structures of the meninges. This is the deepest meningeal layer. Only layer in contact with brain tissue, follows the contours of the brain, covering every sulcus and fissure, permeable to substances in brain extracellular fluid and CSF, central cerebrospinal fluid. Allows for substances to move between fluid compartments, helps balance concentration of different solutes. All right, so it just basically looks like layers of, almost like layers of skin on the brain. That's what it kind of looks like. These are connective tissue that help protect the brain with their gaps in between. Okay. Dura, arachnoid, and pia. This is a closer look at it. Okay. And then we have the ventricles and the cerebrospinal fluid. All right. Ventricles, 4 linked cavities within the brain. Remember, the brain has spaces in it, continuous with the central canal of the spinal cord, lined with ependymal cells, filled with cerebral spinal fluid, CSF, very important. My image. Right and lateral ventricles, first and second, what they're called. in the cerebral hemisphere. Have three regions: anterior, inferior, and posterior horns. You also have a third ventricle, narrow cavity between the lobes of the diencephalon. We saw that when we looked at the anatomy of the diencephalon. And then we have a fourth ventricle between the pons and the cerebellum. And that's continuous with the central canal of the spinal cord. Allows central spinal fluid, cerebrospinal fluid, and ventricles to flow into subarachnoid space. Right, so these are the ventricles, the gaps, spaces in your brain. So cerebrospinal fluid can flow through the brain and also by the spinal cord. Cerebrospinal fluid, CSF. So it's cerebral in your brain and spinal in your spinal cord through the ventricles, through the spaces. So what is cerebrospinal fluid? Since you've probably heard of it mentioned here, and also you probably heard of it before. Many of you probably heard of it before this. Clear, colorless liquid, looks like kind of like water, similar to blood plasma, and it just protects the brain, cushions brain. You know, if anything, any kind of tissue, if you keep it bathed in fluid, it's going to be protected. It's not going to dry out. It's going to be able to move. It's going to be cushioned. Helps maintain constant temperature. Also removes waste. Increases buoyancy of the brain. Allows your brain to not collapse under its own weight. So sort of float, so to speak. Now, your choroid plexuses, another term. There's a lot of terms in this chapter. So I know if you're adding notes to the notes, you're probably like, oh my gosh, I'm like on page 100. This is the choroid plexuses. This is where your cerebrospinal fluid is manufactured. And each of the four ventricles where blood vessels come into direct contact with these ependymal cells. We haven't talked about fenestrated capillaries. They're capillaries with gaps in them. And when you get to the chapter on the blood system, the cardiovascular system, you get the chapter, I believe it's 18, which you won't do this semester, but hopefully next semester. We talk about these fenestrated capillaries, and they have the capillaries with holes in the wall, basically allows for fluids and electrolytes to exit from the blood plasma and enter extracellular fluid. About 150 milliliters circulates through the brain and spinal cord. 750 to 800 milliliters of CSF is produced daily, while the old CSF is removed as the choroid plexuses make new. And this is constantly happening. Your cerebrospinal fluid is being replaced every 5 to 6 hours. Okay. Okay. These fenestrated capillaries, so it's very similar to blood plasma. Okay. Blood capillaries, ependymal cells. All right. Then we also have the blood-brain barrier. Protective safeguard separates cerebrospinal fluid and brain extracellular fluid from chemicals and disease-causing organisms. Right? It's just like it sounds, blood-brain barrier. Consists of simple single-layer squamous flat epithelial cells, known as endothelial cells, of blood capillaries. They're basal lamina and astrocytes. Remember astrocytes, one of the neuroglia of the brain. It has some unique features. They're bound tight by many tight junctions. Remember, tight junctions prevent things from flowing through. So this, in this case, prevents fluid and molecules from passing between them. Influenced by the astrocytes. All right, they limit the capacity of molecules into and out of cell by endo and exocytosis. So substances that easily pass through the plasma membranes are able to pass through the blood-brain barrier. Water, obviously, every part of your body needs water. Oxygen, every part of your body needs oxygen. Carbon dioxide, every part of your body needs to get rid of carbon dioxide. Non-polar lipid-based molecules. You have protein channels that allow for certain other things. Glucose is the primary energy that your brain runs on. Amino acids build proteins, and then we have ions. But mostly your large polar molecules are prevented from crossing that blood-brain barrier. So it kind of acts similar to the plasma membrane of the cell. Barrier is protective, but it can be a hindrance to some medications in the brain. Okay, so this is your typical capillary. Fenestrated capillary allows water and small solutes to move from blood to the extracellular fluid. But we have this blood-brain, in the brain, we have this astrocytes and tight junctions. And brain capillaries limit how much goes from your bloodstream to the extracellular fluid of your brain. Meningitis. Another one of those diseases many of you probably have heard of. Maybe you know what it is, maybe you don't. Maybe you know somebody who's been affected by it. It's definitely life-threatening infection of the meninges. Remember, we talked about the dura, the arachnoid, the pia matter, the layers that cover the brain. Obviously, if those get infected, they're very close to your brain, which would be bad, right? So inflammation causes headache, lethargy, stiff neck, fever. Diagnosis, examination of your cerebrospinal fluid. We can see infectious agents, an increase in white blood cells. Your body's attacking something. Usually bacteria and viruses are the most common causes of this. Viral is generally mild, but bacteria can progress to brain involvement and death, depending on what's happening, right? So infectious meningitis, you have an infection in the meninges of the brain. Okay.